annotation. So I'm giving a different perspective from in civical computational work. And I'm involved in Oxford in the unique department in the Faculty of Interdisciplinary Research, where we're looking at aging, and also I'm heading the biostatistics and informatics in medicine and aging research in the medical school in Oxford. Okay, and briefly I will describe a bioinformatics approach that could be very useful for screenings, and especially for screenings of pluripotency related mechanisms, and also for aging research. And I will talk about ontologies, which are used to formalize knowledge. And knowledge is the basic currency, and for bioinformatics, one typical kind of knowledge are protein interaction networks and protein interaction gene regulation networks. And they're getting very complex. So looking like hairballs, if you reduce the perspective to just describing, for example, pluripotency, that may still be visually handleable, but still it's a lot of information about genes and proteins that are interacting to generate a pluripotent phenotype. The stem cell phenotype previous speaker has also mentioned. And bioinformatics for me is now to reduce this large amount of information to very few relevant findings that can then be investigated in detail. And one example is, in particular, that I've developed a tool where you can use networks, and you can combine the network data with two static data sets describing cells in a specific state. Like in this first step, the state 1 and in the state 2, which may be not pluripotent and pluripotent. And those three inputs, one network and two omics data sets, they could, for example, describe the gene expression. So each of the dots is describing the expression of one single gene, and green is low expression and white is high expression. And if I combine those, and this I've got for two cell states, and then I combine those data, and I get very few interactions highlighted in this network based on what has been described as changing if you're looking at and comparing state 1 and state 2. So the idea is take the network, take a description of one cell state and another, and then you find out that, for example, an interaction is going up from one state to the next. Here, for example, it's a typical interaction for pluripotent stem cells from the NANOT and ESRB. Those are two genes proteins that are stimulating each other. And since NANOT goes up and ESRB goes up from green to red, also the interaction goes up. And that's the basic principle, to find out about interactions, stimulations, inhibitions that are going up or down from one state to the next. So if I've got a low gene expression in the first state, a high one in the second state, then the effect is that the stimulation is starting up. Otherwise, in the opposite case, it's shutting down. And in particular, if there are not much changes, like the gene expression is constantly high, then I can delete that edge. And that way I reduce a large amount of information to a very small amount. And this shutdown and start up can also be strong or weaker. So, and it's based on a network that I've assembled, and we are still updating it, describing pluripotency, stemness, embryonic stemness. We've got a lot of publications, about 200 publications 2 years ago, and now it's about 400 publications on which we base that network, also small molecules included, and so stimulations, interaction, inhibitions related to pluripotency in mouse. And the tool we are using is this presence tool, which does a differential comparison. So we can take time series, different conditions, or even different species, to then find out about the most prominent mechanisms that are suggested by the complementary data, by the differential data. That's complementary to tools that are highlighting active subnetworks. Some people of you may have seen those tools that are also very popular. But this is kind of complementary, and in particular, we are finding small subnetworks of interacting genes, like KLF4 and ESRB in this case, and not larger subnetworks. So it's important to have a few links that you can investigate, and there are also papers that are kind of supporting this view that functional information within gene networks and also protein interaction networks typically concentrate only very few interactions. And those are the very few interactions that we are finding with our method. And the typical use case is that here we've got the network is carrying pluripotency again, and now we are placing data on the network, describing the steps from a differentiated cell, like a fibroblast, that is driven towards a pluripotent state. So on the left you've got the gene expression values of the fibroblast, on the right side you've got the gene expression values of a pluripotent cell. And this IPS, so induced pluripotency, can now be described in its effects in the network. So interactions are going up and down, and we can take a slider, so you can focus on the very most important interactions. And that's very relevant because you can now visualize the steps of a fibroblast to a pluripotent IPS cell in a kind of movie. Like here, we have taken a time series that has been published by Zamari Tirani about 2 years ago, and it shows that in the first step, so it's a 7-step time series, in the first step, interactions around a protein called OCT4 are going up, then around NANOC, still around NANOC in the next step, and then around ESRB, again around NANOC, and finally around SOX2. And SOX2, OCT4, and NANOC are 3 of the very typical pluripotency-related genes. And this movie describes this IPS process. And I'm going back, and if I'm going forward again, we can track the hypothetical or the hopefully real actions that are going on in that experiment of transforming a fibroblast into a pluripotent cell. And that's of course very interesting because once you have got small molecules also included in the network that are describing the small molecule effects, you may try to emulate that process by small molecules. This one is IPS is a more or less genetic process, and nobody up to now has come up with small molecules that can accomplish the same feat. And that would probably be very relevant and interesting because small molecules are usually easier to handle, and they may be selected for not being oncogenic and similar attributes. Okay, so bioinformatics is here used to first describe reprogramming of a fibroblast to an IPS cell, and then to suggest small molecules that may do just the same thing, which then gives rise to screening approaches, where you would say, okay, I take those small molecules in the first step, another set of small molecules in the second step, and so on. Yeah, and another study with the same network, we've been looking at the transition of an embryonic stem cell to an epiblast stem cell. So 2 kinds of stem cells. I also mentioned the last talk, if I remember correctly. And this is the epiblast stem cell is a kind of slightly differentiated pluripotent stem cell. And you can see that here the network boils down basically lots of actions around ESRB. So there's not much action around the standard players, OCT4, SOX2, and NANOC, because it's going from pluripotent to pluripotent. So not much happening there. But around ESRB, there are lots of stimulations, that's the arrows, that are being shut down. That's a green color. But the gene itself doesn't change much. However, then if we take a look, not after 12 hours, like here, but after 48 hours, you will see that this gene is actually going down rapidly. So that's a nice thing for a bioinformatician, that he does a prediction here about what the gene will be doing in the future, and the first experiment the colleague at the Max Planck did actually then immediately confirmed ESRB is going down later on, which is quite nice. Okay, and in the second part of the talk, I would now like to describe ideas about, coming actually from research in computer science and also in philosophy about ontologies. And ontologies are ways of having a formal vocabulary, a well-defined vocabulary, and also having encoding knowledge about how the terms relate to each other, and which things are parts of other things, for example. And ontologies usually start with continuance and occurrence, so the world is divided into things and happenings, events. And actually, what we did just a few minutes ago is taking three continuants and transforming that into a set of occurrence by inferring that there is an interaction startup, which is an event. It's an event based on those static data. And in this work in progress, we are now trying to put this into context. For example, we've got an interaction of ESRB and nano, as you've just seen. And this is a mechanism, and it is part of a subnetwork of other interactions around this ESRB and nano. So we are trying to put this all into a hierarchy, something like the gene ontology hierarchy, if you happen to have heard about that one. And we can do that also for other molecules, for example, octodine-claudine, and subnetwork interactions around it. And then we can put it into a more complete picture. So knowledge representation now means that we've got specific knowledge, and this is related to more general knowledge because the interaction is a mechanism, is a process, is an occurrent, there's participants of it, and so on. And we can also be more specific here that we say the network of mechanisms relevant for the IPS has a part that is a network of mechanisms relevant for epithelial cells, for tight junctions, for octodine and gem-related interactions, for the interaction of octodine-gem. So there has part relation from here to here that's based on literature knowledge. So the literature tells us this. They've recently demonstrated IPS is actually part of the IPS process is that an epithelial phenotype forms, which has tight junctions and so on. So the idea is to have a hierarchy of knowledge, where you can ultimately do calculations. And that's becoming interesting once you have got, for example, several parts. Say this network, subnetwork interaction of octodine and gem has two parts. Octodine and gem, octodine and DJB1. And also this network mechanism relevant for tight junction has two parts. And then you can take papers describing processes related to, for example, IPS. You see there's a tight junction, and you can infer that those things must also be happening because those are parts of the network of mechanisms relevant for tight junctions. So you can, so once you have a paper that is describing something in a figure with tight junctions, you can conclude the other things. Of course, biology is dirty; it will not always work. But if there's reasonable confidence, you can conclude those things. And then you can imagine that you've got another paper describing another reprogramming process, or also describing a cancerous process. Well, you also have mesenchymal and epithelial phenotype changes. And if, for example, that paper just describes interactions of octodine and gem, you can infer this. Maybe another paper just describes a molecular process, or maybe a bioinformatics analysis just gives you the molecular processes. And now, based on the inferred marks, we can actually do a comparison. Imagine you would just have the black marks. You could not really compare the processes. But based on the ontology and on the marks that you have inferred, you can now say, okay, they are very similar here. And this is one of the values of doing such ontologies, and encoding knowledge, and making processes, events comparable, like reprogramming MET, or maybe an MET happening two different kinds of cancer, for example. And you can have bioinformatics, high throughput-based knowledge of individual genes in the same hierarchy as more more general things like phenotype descriptions, altostructure descriptions of like junctions of specific cell components. And this is actually motivated by a similar approach done in 2009 based on human disease, where they are describing human diseases, also using this kind of ontologies. And we have got a preliminary ontology of mechanisms, where we can include bioinformatics data, and actually these expressions I've been talking about is just about finding out that an interaction outputting jump, for example, is up. So this matches very nicely. And you can also have altostructure, cell components described, and you can put it all into this hierarchy. And we've also got a hierarchy of continuance of cell phenotypes, which follows a kind of similar set of rules. Again, you can have bioinformatics knowledge and manual curation, and we can even put both ontologies together. So there's a state where the cell phenotype is described, another state, a third state, and we can describe the changes from one phenotype to the next in an ontological scheme, which is an entity quality scheme. That those details don't matter so much, but they connect to this epigenetic landscape, this volume landscape. You've seen that in one of two talks, that cells are actually in a quasi-potential landscape. There are clusters of cells and another cluster of cells, and cells change usually in the trajectory towards of least resistance. And the mechanisms, they are the changes from one of those clusters to the next one. So this has some nice implications for describing scientific accomplishments. So this is actually was still a project looking for funding. So we would like to encode data based on publications and omics data, and propagate annotations, calculate information content, run clustering and similarity search based on that. Similarity search I've motivated in one of those slides. And then we could find out, for example, that reprogramming MET, this cancer-like process, is also close to breast cancer MET. And we would like to find small molecules that influence reprogramming. Of course, you can imagine you can also do the same things for aging-related data. We would actually like to crowdsource molecular aging mechanism data. There are some people who are interested in this, like Woodkish or Magalas, like Langin maybe. And then we would like to identify small molecules that slow the aging process. So we are now not comparing fibroblast to iPS cell, but disease to healthy cell, old to young cell, and so on. And we would like to use high throughput data to find out about the different mechanisms, and then try to find small molecules that may be mimicking that. Yeah, that's I'm coming to the end. Actually, the last slide, I'm organizing workshops on a regular basis. One on also symposium systems biology and bioinformatics in aging research. We will have focus on ontologies next time. This will be in September 2013 actually, sorry that's a typo, 2013. And two of the speakers will be supported by the aging department, and past events have also been supported by pharmaceutical companies and by SENS. And also we have a bioinformatics and stem cell satellite workshop in April 2013, and there we also have very interesting speakers that supported by the DFG. And I would really welcome contributions by you. And that's the final slide. So I'm just in time. Thanking my graduate students and collaborators, and I'm happy to take questions.
I thought that was wonderful, and that the presentation of that and information, it looks superbly helpful in a number of different areas. I'm just wondering, is there plans to bring this towards education or education curricula, or is it something that you envisage going into universities at some point?
I'm actually using this in my lectures. So I'm running a bioinformatics lab in the summer, and the students are working for about 5 weeks with tools for network biology, and in the final session they are working with the 6-process tool, trying to find out mechanisms themselves.
And as a learning tool?
Yes,
yeah.
That's with another learning tool.
Perhaps a similar question for the many amateurs online who have algorithmic and programming skills. Is there something to do here with distributed forces, or is it something?
Yes, we would like to make this a crowdsourcing approach, this the ontology development. And Daniel Victor is very enthusiastic. He created this Enigma on the net, where we would like to do crowdsourcing of this ontological knowledge. And the idea would be that it ultimately is a resource where you can place all the aging-related information from publications into one single integrated database. So of course also a huge task, but I think that such a huge task can maybe realizable by crowdsourcing. Like Wikipedia is also a huge task which was realized by crowdsourcing.
There is already someone in France, Bruno Cormont, you know, Bruno Cormont, who has made a database for aging. He has made a small startup.
Please let me know. Give me the link to that, and I would be interested also in getting in contact